My name is Emily Pitzer. Thank you for inviting me to speak on this topic today. I will be going over linkages between AOP key events and neurotoxicity assays, specifically providing an example looking at glutamatergic modes of action. And I want to disclose that the views presented here are my own and do not necessarily reflect the views or policy of the U.S. Environmental Protection Agency. Great progress has been made over the past 40 years or more in understanding the hazards of exposure to a small number of neurotoxicants. Overall, there are many chemicals on the market, with which only a few have been extensively studied, however. In addition, new patents are being made each year for new chemicals coming to market. The current methods, which predominantly focus on in vivo animal data, can be both labor and cost extensive, as well as require a high level of expertise for both their interpretation and implementation. It is clear the need for alternative methods to effectively and efficiently screen chemicals for neurotoxicity or other hazards in general is necessary. Several methods have been implicated to date to try to tackle the challenge of studying chemical hazards for impacts on the brain. In addition to in vivo assessments, methods in vitro to in silico are being implemented. However, there is a need to understand where gaps in these alternative approaches still remain and where current overlap and consensus is found. We'll be focusing on the use of AOPs, or adverse outcome pathways, that are employed using data from various modalities from in vivo to in vitro results. The EPA New Approach Methodologies, or NAM, in vitro assays are being employed to assess chemical hazard for neurotoxicity, which are focused on covering some of the key neurodevelopmental processes in the brain. This includes functions such as network formation, which is measured by microelectrode array, or MEA, as well as synaptogenesis, proliferation and growth, and apoptosis, which can all be measured by high content imaging. But how does all of this fit into our work focusing on mode of action based studies or the development of adverse outcome pathways, or AOPs? Our group at the EPA are working towards developing these AOPs for adverse health outcomes that are relevant to risk assessment or regulatory decision making. In brief, the AOP framework describes a series of events that are linked at different levels of biological organization, such as from the cell, the tissue, to the organ, that lead to an adverse health outcome in an organism following exposure to a stressor, such as a chemical. Both in vivo and in vitro findings can map onto the AOP framework at the cellular key event level. And we hope to use data that we are generating here to build out these frameworks and aid in our understanding of neurotoxic effects, especially in developmental models. So we're trying to fill in gaps in our understanding on how toxic insults can lead to adverse effects in the brain using proteomics in particular. First, I want to explore where the in vitro NAMs do align with current AOPs. There are many identified in the literature, but our group has done a recent systematic search of neurotoxicity and developmental neurotoxicity AOPs on the AOP wiki site in particular. And we were really focused on examining those that were with an endorsed OECD status. So the AOP Wiki is a web-based repository containing qualitative information for AOP frameworks and is an effort coordinated by the OECD, or the Organization for Economic Cooperation and Development. So this OECD status, it details the inclusion within this larger knowledge base project as part of this AOP development work plan, which is why we really focused on these identified AOPs in particular. This review I'm speaking about was conducted in spring of last year, so a few additional AOP models have since been included and added to the wiki site, but they're still limited in number overall, as well as not fully encapsulating all potential impacts to the brain or modes of action that could really be targeted. From our review, we did find alignment for DNT new approach methodologies and key events involving processes such as apoptosis, network formation, as well as growth and synaptogenesis. Noting this importance in focusing these key neuronal processes for DNT and neurotoxicity studies. However, this does highlight that there are still several key neuronal processes that are currently not being observed among these key events or in the current AOPs on the wiki site. We did find that the key events that align with the in vitro assays, many of which are found when perturbing these major neurotransmitter systems in the brain. Here I have a network of AOPs focusing on molecular initiating events targeting the main excitatory and inhibitory neurotransmitters of glutamate and GABA. Events are shown here in the yellow boxes. Those highlighted in pink are found to link to current in vitro DNT assays. And as you can see, there are several popping up in this diagram. Our group has then focused on targets that could impair these key processes to examine for further effects and aid in corroborating in vivo to in vitro results. See how these key biological processes in the brain can be affected by chemical insults. We are measuring proteins. Specifically, we are measuring proteins using a global proteomic analysis, so this is using a non-targeted approach. The work presented here is done in animal models, but we have related studies that are being done in vitro to examine comparisons between proteomic results. Both the in vivo and in vitro findings can map onto our AOP framework at the cellular key event level, and we hope to use this data we are generating at the EPA to build out these frameworks to aid in our understanding of neurotoxic effects in both adults and developmental models. So we are trying to fill in gaps in our understanding of how toxic insults can lead to adverse effects in the brain. Here we are utilizing canic acid as a chemical probe. So canic acid acts via the AMPA or kainate glutamatergic receptors, activating them. This is a pharmacological agent that can exert its effects through excitotoxic or epileptic properties, triggering various neuronal signaling pathways. So we really aim to use this chemical probe to observe the effects on the proteomic signature following exposure. The study was done acutely in adult male Long Evans rats, examining doses that do not produce myoclonic seizures, which canic acid has been used as a pharmacological tool in other studies. We collected tissues for proteomic analysis at 3 or 24 hours following exposure, performing region-specific dissections of the brain. Histology was also performed at similar time points as well as 48 hours after exposure. Tissue samples were then processed and analyzed on the Orbitrap mass spec and further assessed in Proteome Discoverer. Those results were then applied to bioinformatics software, which is what I'll be showing you today. Due to time, I will be focusing on effects in the hippocampus for canic acid exposed animals compared to controls. Before I dive into our pathway analysis, I first want to highlight the histology work that was done by our colleagues at NIEHS. This H&E staining was done after exposure in the 6 milligram per milliliter canic acid dose, and these are at sub-seizure dose levels where we didn't observe any microscopic lesions, including within the hippocampus. However, lesions are observed at dose levels which do produce seizures, and importantly, this was shown in the hippocampus as I have shown here. In terms of our proteomic signatures, we first assess impacts on biological pathways identified from our data set. There are a limited number of significantly altered proteins overall, but what we were able to see in several pathways predicted to be impacted following this exposure. Here are the top 10 pathways at both time points. At the 3-hour time point, we did observe several pathways pertaining to mitochondrial functions. And at both time points, we saw pathways involved in biosynthesis processes, such as controlling degradation or production of a variety of molecules, as well as cellular processes, such as cellular movement, assembly, organization, growth, and survival. And lastly, among the top pathways at the 24-hour time point, we also saw several relating to neuronal and intracellular signaling. These are overstretching examples of what these pathways are doing, but just to get an idea of what we were observing at these time points. The top pathways, several others are identified in our analysis. Of which related to glutamatergic systems or related processes, which we really wanted to further scrutinize due to canic acid's molecular target of this system, which is shown here in this table. Several relate to activity of glutamatergic receptors or the degradation or metabolism of said molecule, as well as actual signaling at the synapse. These pathways are all identified at the 24-hour time point, analysis, but are not found within the 3-hour time point, except for synaptogenesis signaling, which is seen at both. Synaptogenesis is a process that is captured by the in vitro battery, but it is unclear if these other pathways identified here would be seen by neuronal signaling related in vitro assays that are currently developed. It's also understood that chemicals targeting and activating glutamatergic systems could elicit excitotoxicity, which in turn could trigger various inflammatory processes, cytokine signaling, as well as potential cellular death mechanisms. Canonical pathways relating to cell death, survival, or stress were examined next in our IPA results, of which several pathways were found to be identified at both time points. Several are specific to mitochondrial stress or mitophagy, as well as inflammatory signaling pathways. And although many are implicated in cellular stress or inflammation here, they do not necessarily result in cell death and thus may be missed by the current in vitro assays, at least at the dose levels used here. Thus, it's really noting the potential gaps to focus on for further development of alternative methods or areas where our in vivo testing may still be needed. IPA, we also examined additional pathway analysis software for further understanding of this data set. All of these tools can differ greatly in the type of functional information they tell you, such as Go category analyses to topological information, such as network level information. And what I've shown you so far is from IPA, which generally sits in the middle of these domains. However, we wanted to further explore additional tools on both of those levels that are more suited for functional information like David and KEGG, as well as those that add more network level information like Cytoscape. It was assessed in David, which is a bioinformatic tool focusing on functional annotations. Autophagy and apoptotic processes were among the top terms enriched in our data set, as well as enriched terms relating to the mitochondria, which really further corroborates our effects that we observed in IPA. We also gain a deeper understanding into terms of organelles and locations within the cell of interest, such as the Golgi or the cell membrane, where a chemical could be eliciting its effects. By examining these proteomic results through multiple tools, we really can start to see important biological functions that overlap, as well as gain confidence in these effects. We examined Reactome to understand biological pathways impacted and observed concordance among our analysis tools. At the 3-hour time point, we see several RHO or RHO GTPase cycle pathways identified, which was also observed in our IPA predictions. These are important signal transducers in pathways that regulate cell migration, proliferation, survival, and death, and are crucial for maintenance of normal tissues and can contribute to disease progression. Within Reactome, we also observed G-beta signaling and glutamate metabolism pathways, which could corroborate, again, impacted pathways found in IPA. So overall, we're seeing these additional tools gain further confidence in the pathways we are identifying, as well as additional functions to help build out our molecular framework. We examined our data set within Cytoscape to begin to understand the network level effects following our exposure in vivo. Each network is shown at a given time point, only including the molecules that generated this network structure, with downregulated molecules are shown in green and upregulated in red. We observed several molecules at both time points involved in neurological diseases and neurotoxicity. Also, these molecules are implicated in effects in trafficking and degradation, as well as impacts on cellular signaling. In general, we can understand further how these molecules are interconnected, but really additional studies are needed to explore how these connections then fit within to our adverse outcome pathway. In conclusion, we did observe an altered proteomic signature following acute canic acid exposure in our rat model utilizing the sub-seizurogenic doses. We saw implications for processes involved in mitochondrial and cellular stress, as well as altered neuronal signaling. Some of these observed effects may also correlate to measures that are part of the new approach methodologies that are being employed to assess neurotoxicity, such as measures relating to network function or synaptogenesis. However, there clearly are some potential areas to expand these current assays, such as observing more changes in inflammation, cellular stress, as well as records for neurodegeneration that cell death alone may not capture. Overall, this proof of concept study is being expanded to study more test chemicals and further explore effects including neurodevelopmental neurotoxicity. I would like to thank you for your time. I also wish to thank all the collaborators that were part of this project. And please let me know if you have any questions, and always feel free to reach out to me after this for any further discussion. Thank you.